What if we had the power to cure all genetic disease? To eliminate sickle cell anemia, Huntington's disease, and even a predisposition to cancer with a single precise edit to our own DNA? What if we could use that same power to design our children, to make them taller, smarter, more athletic, or immune to viruses? This is not science fiction. This is the stunning reality of a revolutionary technology called CRISPR. And Walter Isaacson's brilliant book, The Codebreaker, is the definitive story of the scientists who unlocked this awesome power, and the profound philosophical and ethical questions it raises for the future of the human race. This is more than the story of a single scientific breakthrough. It is a gripping tale of collaboration and competition, of Nobel Prize-winning triumph and deep moral anxiety. It's a story about the moment humanity gained the power of the gods, and the terrifying, exhilarating, and essential process of figuring out the rules. This is a journey to the very frontier of science, ethics, and what it means to be human. The humble origins of a revolution. The story of the greatest biological breakthrough of our time does not begin in a futuristic, high-tech lab. It begins in the most humble of places, a yogurt factory. For years, scientists working with bacteria to make yogurt and cheese had been puzzled by a strange feature in the bacteria's DNA. They found these weird repeating sequences of genetic code, separated by unique spacer sections. They gave this strange pattern a clunky name, clustered regularly interspaced short palindromic repeats, or CRISPR. For a long time no one knew what it was for. It was a biological curiosity, but a handful of brilliant and obsessive scientists, including a Spanish researcher named Francisco Mojica, began to unravel the mystery. They discovered that the unique spacer DNA in between the repeats was not random. It was a perfect match for the DNA of viruses that attack bacteria. This was the aha moment. CRISPR was an ancient adaptive immune system that bacteria use to fight off viruses. Here's how it works, in simple terms. When a virus attacks a bacterium, the bacterium, if it survives, will grab a snippet of the virus's DNA and weave it into its own CRISPR region, like a genetic mugshot. It's creating a memory of its attacker. Then, it creates an RNA copy of that mugshot and attaches it to a special search-and-destroy enzyme, a protein called Cas9. This little complex then patrols the cell. If that same virus ever attacks again, the RNA guide will recognize its DNA, and the Cas9 enzyme will act like a pair of molecular scissors, finding the matching viral DNA and precisely cutting it, destroying the invader. This is the brilliant, beautiful, and ancient system that nature created. The great genius of the heroes of our story was not in inventing this system, but in realizing that this bacterial immune system could be hijacked and repurposed into a tool for editing any DNA at all, including our own. The Codebreaker, Jennifer Doudna. The central character in this epic story is the American biochemist Jennifer Doudna. Isaacson paints a portrait of a brilliant, driven, and deeply humane scientist. The key moment comes when Doudna, a professor at UC Berkeley, meets a brilliant French scientist named Emmanuel Charpentier at a scientific conference. They decided to collaborate. The story of CRISPR is not just a story of a single heroic discovery. It is also a gripping and often messy story about the very human world of science, a world filled with intense competition, brilliant collaboration, and battles over credit and patents. While Doudna and Charpentier had shown that CRISPR could be programmed to cut DNA in a test tube, another brilliant young scientist at the Broad Institute of MIT and Harvard, named Feng Zhang, was working on a parallel track. This set off one of the biggest and most bitter intellectual property battles in the history of science. As the power and potential of CRISPR became clear, Jennifer Doudna began to experience a profound sense of anxiety. Her role began to shift from being purely a scientist to becoming a moral and philosophical leader. The turning point came in a dream. She dreamed that she was in a room with Adolf Hitler, who had a notepad, and was asking her to explain the uses and implications of the technology she had just created. She woke up in a cold sweat. In that moment, she understood, on a deep visceral level, the terrifying dual-use nature of her discovery. The same tool that could be used to cure horrific genetic diseases could also be used for horrifying eugenic purposes. This dream was a wake-up call. Doudna realized that she and the other scientists in the field had a profound responsibility, not just to continue their research, but to lead a global conversation about the ethics of this new power. She began organizing conferences, bringing together scientists, ethicists, philosophers, and policymakers to grapple with the profound questions that CRISPR raised. to grapple with the profound questions that CRISPR raised. The central ethical dilemma is the distinction between two different kinds of gene editing. The first is somatic gene editing. This is like performing a therapy on a single consenting patient. You would edit the genes in, for example, the blood cells of a person with sickle cell anemia to cure their disease. These changes would not be passed on to their children. Almost everyone agrees that this is a wonderful and ethical use of the technology. But the second kind is germline gene editing. This is where you would edit the genes of a human embryo, a sperm, or an egg. These changes would not just affect the resulting person, they would be passed down through all future generations. This is a permanent alteration of the human gene pool. This is where we enter the world of designer babies. You could, in theory, edit the germline not just to remove a disease gene, but to add genes for desirable traits like height, intelligence, or athletic ability. This is the Pandora's box that Doudna realized she had opened. Where should we as a species draw the line? Who gets to decide what is a disease to be cured and what is a trait to be enhanced? And what would it mean for society if the wealthy could afford to genetically enhance their children, creating a new biological class system? These were the terrifying and essential questions that Jennifer Doudna, the codebreaker, was now forced to confront. The Pandora's box is open for several years. The global scientific community, led by figures like Doudna, held to a fragile consensus. They agreed that while somatic editing for diseases was a noble goal, germline editing in humans was a line that should not be crossed, at least not until there had been a broad public debate about the safety and ethical implications. And then, in 2018, a young Chinese scientist named He Jiankui shattered that consensus. He shocked the world by announcing that he had done it. He had used CRISPR to edit the genes of human embryos and that two twin girls, Lulu and Nana, had been born. He claimed that he had edited a gene called CCR5 to make the girls resistant to the HIV virus. The global scientific community reacted with immediate and universal condemnation. He had been reckless. He had been unethical, he had performed a dangerous experiment on human children without any clear medical need, as there are much safer ways to prevent HIV infection. He had broken the fragile moratorium. The story of He Jiankui is a chilling and cautionary tale. It showed that no matter what rules or guidelines the scientific community agrees to, the technology itself is now out in the world. It is relatively cheap and easy to use, and there will always be rogue actors driven by ambition or a misguided sense of heroism who are willing to cross the ethical lines. The Pandora's box of human germline editing is now officially open. We are now living in the future that Jennifer Doudna had nightmares about. The COVID-19 Coda and the Future of Humanity. The book ends with an unexpected and powerful real-time demonstration of CRISPR's incredible potential for good. As Walter Isaacson was finishing the book, the COVID-19 pandemic swept across the globe, and the scientists who had been at the center of the CRISPR story immediately pivoted their labs to fight the new virus. Jennifer Doudna's lab and others like Feng Zhang's used their deep knowledge of RNA and CRISPR to develop new, rapid, and cheap diagnostic tests for the virus. Others began to work on using CRISPR as a potential antiviral agent, a way to find and destroy the virus's genetic material inside our own cells. And of course, the revolutionary mRNA vaccines that were developed in record time were themselves a product of the same revolution in our understanding of RNA and genetic code that had led to CRISPR. This final chapter is a powerful reminder of the incredible promise of this technology. In the face of a global crisis, the heroes of the CRISPR revolution stepped up. It showed that this tool, which can raise such terrifying ethical questions, is also one of our most powerful weapons in the fight against disease. The ultimate message of the Codebreaker is that we are at a pivotal moment in the history of life on Earth. We have, for the first time, gained the ability to consciously and deliberately edit our own evolution. This is a power of unimaginable consequence. The book is not just the story of a scientific discovery, it is the story of the beginning of a new era. It is a call to action for all of us, as citizens, not just as scientists, to engage in the most important and most difficult conversation of our time. The code has been broken. Now, we have to decide what to write with it. If you believe that we have a shared responsibility to grapple with the future of our own species, then you are in the right place. Welcome to Geeky Philosopher. Subscribe and let's continue this essential inquiry together.